Okay, my objective was the immune response to tumors, and Abbas had a great section in cellular and molecular immunology, and there was also a really great section in the biology of cancer, second edition. Those are going to be my two primary sources. Prior to the 1950s, there was a lot of anecdotal evidence that there was immune responses to cancer. For example, whenever tumors were excised, they were found to be surrounded by the mononuclear leukocytes, for example, T cells, NK cells, and macrophages. And when those infiltrates were present, you would have a good prognosis, but when they were absent, it would actually predict a poor prognosis, and that was especially true in melanoma, breast carcinoma, and carcinoma of the colon. In the 1950s, there were some really cool experiments, and I want to go over them really quick. First, we got an inbred mouse. It looks happy because everything that's inbred looks happy, I think. That's what my parents told me. Then somebody took some methylcholinethrin, I'm really not sure if I'm saying it right, but we'll just call it MCA, and they painted it on the mouse. This is a known carcinogen, and it caused the formation of a tumor. So the next step was they cut the tumor out of the mouse. So again, this is my inbred mouse, and this is my syngeneic mouse. So they cut the tumor out of the inbred mouse, and then they implanted it into a syngeneic mouse. So the point of this is there was an initial immune response to the tumor in the inbred mouse, and then they took the tumor out and they allowed that was like a vaccination against this tumor. So they let the tumor grow in the syngeneic mouse. And so that's what I just said, tumor grew in the syngeneic mouse. Then they cut the tumor out of the syngeneic mouse and they replaced it into the inbred mouse. And by the way, as a side note, I drew these mice all by myself, in case there was any question. So once they put that tumor in that mouse, the tumor just started to be eaten up by the T cells and everything. Well, the tumor died. We can't say that it was the T cells at this point. But wait, before it completely died, there's a little bit left. We went ahead and we decided to cut that out and put it back in to not the same syngeneic mouse, not necessarily the same, but into another syngeneic mouse. And the cancer begins to grow in this mouse, and we decide to take some T cells, these are T cells, and we put them in here, and that causes the tumor to go away. So that proved that there was a T cell mediated mechanism that was fighting tumors all the way back in the 1950s. And that's what it said right here. It said to prove that the T lymphocytes killed the tumor, infrared mouse T lymphocytes were transplanted into the syngeneic mouse. The tumor died in the syngeneic mouse. So that was basically a 1950s experiment. Okay, so here's an overview basically of what I'm going to talk about, broken down into three parts. Part one is that tumors do stimulate an adaptive and an innate immune response. Part two is the immune response fails in some cases, and that in part three, new treatments seek to basically superactivate the immune system or to support those parts that are failing in whatever way. Okay, so we know that tumors produce antigens. So there's two different kinds of antigens that tumors can produce. The first one is called tumor-specific antigens, and the other one's tumor-associated antigens. A tumor-specific antigen is unique to the tumor, and no other tissue will actually express it or produce it in a normal, active, live human being. A tumor-associated antigen is produced in normal cells, but it's produced in a strange way in the tumor cells, or in other words, it is expressed aberrantly in the tumor cells. So what I have on this slide is actually basically how they discovered which antigens were actually tumor antigens. So basically I'll explain it like this. Have this person, and the person has a cancer. Cancers are always red, right? And within that cancer there were a bunch of T lymphocytes, T lymphocyte infiltrates. What we're going to do is we're going to take a petri dish and we're going to take those T lymphocytes and we're going to put them in the petri dish so that we have harvested the T lymphocytes. Next thing we're going to do is we're going to take a next-gen DNA sequencer. So that star is a next-gen DNA sequencer, and we're going to take a biopsy of the cancer, and we are going to put the cancer in the next-gen DNA sequencer. And we're not going to sequence the DNA. We're going to sequence something called the cDNA. And cDNA is basically if I take the RNA that's being expressed and I turn it back into DNA, that's called cDNA or clone DNA. So I want to know what proteins the tumor is making. So I find out by analyzing the cDNA in NextGen Sequencer. And once I find out, I take and I grow those proteins. I grow those proteins in a different cell. So I have different cells here. They're not the same tumor cells, but they're different cells. And I grow those proteins. I cause only those proteins to be expressed. So of course, if you know anything about this, then you know that I'm simplifying things. But then I take these lymphocytes and individually, if protein 1 is in this plate, then I put the lymphocytes in with protein 1. Then in another plate, I'll have cells that are expressing protein number 2. So protein number 2, and I put some of the lymphocytes in this plate. And I continue doing that until I've checked every single individual protein that's expressed by the tumor cDNA, and I see which ones are having T lymphocyte reactivity. And so that's what I said. I have said that a novel method to discover tumor-specific and tumor-associated antigens is called Sareks. You obtain the CTLs and CD4 T cells from the infiltrates. You make a cDNA library from the tumor. You express each cDNA in another cell line. You incubate the cells with lymphocytes, and then you detect you have an assay for which proteins are causing lymphocyte activity. And this is called serologic analysis of recombinant cDNA expression, or Sareks. And I put this little piece of information here because it doesn't fit anywhere else, so to speak. And basically is that antigens can be presented on MHC1 or MHC2 via dendritic cells. And so you can get activation of both the CD8 and the CD4 T cells. And we're going to talk about this cross presentation by the dendritic cells in a minute. So I'm saying that tumors induce a specific immune response, and they can do that because antigens can be presented via dendritic cells or via the actual tumor cells on MHC class 1. And then we also know that animals, when they are immunized with mutant p53 or with mutant RAS, they're able to reject tumors that are expressing those mutations. The antigenicity of specific p53 and specific RAS mutations has not been as strong in humans as it has been shown to be in animals, and there's a lot of theories on why and how that can be changed. And now here's the meat and potatoes of part 1, that tumors induce immune responses. So tumors would not produce an immune response if they did not produce some kind of antigen. And so that's the take-home point. There has to be an antigen to produce an immune response. And those antigens are broken down into different types. The first type, mutated oncogenes or tumor suppressors, and we just talked about this, RAS, p53, also the p210, and the BCR-ABL rearrangement product is the p210 product. 10% of human carcinomas have a RAS mutation. 50% have a p53 mutation. And so if we can make the immune response more specific and more robust against a p53 mutation, then we could eliminate a lot of tumors. The next class is unmutated but overexpressed oncogene products. When this is overexpressed, it's strongly associated with certain types of tumors of the breast, and there's actually antibodies that specifically bind to HER2/neu, and when they do, it's great chemotherapy. Now there are some gene products that are normally silent in almost all tissues. These are specifically called cancer testis antigens. And when you hear cancer testis antigens, what you want to think are antigens that are typically produced only in gametes within the testes or within the trophoblast of the placenta. So you think placenta or testes, and you think gametes or trophoblasts. So these antigens usually don't ever see the light of immune daylight. So let's just take a few seconds to talk about cancer testis antigen, because it's sort of I'll just say interesting because I don't think it's high yield. In fact, I don't really think much of any of this is really high yield. I just think it's interesting. So the first point is that it's produced in either the testes or placenta, either via gamete or trophoblast, which is what I just said. These are antigens that are found on several different kinds of tumors. The genes for most of these antigens are on the X chromosome. Okay, these are usually unmutated proteins, so they're not being expressed because they're mutated. They are not mutated, but they're actually just being expressed for no good reason at all, and their function is either not known in most cases and typically not necessary for the tumor. Now, a lot of these are being tested in different vaccine trials, but here's the double-edged sword. They make for great targets because they're not your immune system's not going to attack your balls, at least I don't think. And the trophoblast is usually in a protected environment and secretes all kinds of anti-immune chemicals. So it makes for a great target for vaccines. However, the double-edged sword is that these are not necessary for tumor survival or for the cancer phenotype. And so if the cancer mutated something that caused these to stop being produced, then the vaccine would be ineffective. However, in all of the tumors where that mutation did not occur and they were being expressed, those we could still vaccinate against if those vaccines end up working. They're in clinical trials. So the next one I have highlighted in red because it's kind of a surprising thing, and that's the first thing is that its non-oncogenic proteins are overexpressed, and that's not really a huge surprise. The surprising thing is that we actually get a CD8 CTL and a CD4 positive T cell response to this gene. Here's why that's surprising. So here I've drawn a perfect picture of the epidermis. And in red, I've highlighted two melanocytes. This tyrosinase that is being produced by the melanocytes is important for the production of melanin, and it's expressed all the time. And when it's expressed all the time, it never induces an immune response. However, in this case, now I have a melanoma. You know it's a melanoma because first of all, it's red, and second of all, it's big. And thirdly, it's like in the same place where the melanocyte used to be. Which brings us back to the ABCDE rule. It's asymmetric. It has border irregularities. There are obviously and always have been color irregularities. If this were a pencil, we could say that the diameter is greater than the width of a pencil. And E, we saw that it is enlarging. In fact, it used to be this size. You see when you get melanoma, you get these tyrosinases produced in high quantities, and from that with a combination of the inflammatory reaction, we all of a sudden have tyrosinase being presented to the CD8 and CD4 T cells. And they actually do mount an immune response against this enzyme. Tyrosinase is another one of those antigens that are being explored in clinical trials for its potential as a vaccine. And specifically a vaccine against melanomas, which is like every young girl's pipe dream, I guess. It seems like every young girl wants to get a melanoma. The next thing we have is virus products, and now typically if we're talking about viruses and cancer, we're talking about most of the time DNA viruses. So HPV with the E6 and E7 protein, Epstein-Barr virus with the EBNA protein. In fact, the only well-defined retrovirus that causes cancer is HTLV1, so everything else we're thinking of DNA viruses. And the take-home with these, especially with the DNA viruses, is that they're fairly harmless if you have a normal immune system. Now, this isn't a universal truth, but we do know that the amounts or the cases of squamous cell carcinoma of the cervix and stuff actually increases in HPV-infected individuals. It increases during immunocompromise, and so we have the immune system keeping that in check for the most part. And lymphomas due to Epstein-Barr virus are commonly seen in people who are HIV immunocompromised. So the next type of antigen is oncofetal. We only have 3 left. Oncofetal antigens. The 2 main characterized oncofetal antigens are alpha-fetoprotein. The other one is called carcinoembryonic antigen, or CEA. This is also known as CD66. Now the key with both of these is that they are usually expressed only by fetal tissue, except during times of inflammation. So during times of inflammation, the liver actually does increase the production of alpha-fetoprotein. And it also increases the production of this carcinoembryonic antigen. Cirrhosis of the liver increases alpha-fetoprotein. So the point here being that we can't use these as targets for therapy, but they can be used to monitor specific antigens. So it's basically like using the ESR or the CRP to monitor autoimmune processes. You can use these 2 proteins to monitor several types of cancers. So the next one is really interesting. This is glycolipids and glycoproteins. So here's the idea. You have a cell, and typically that cell is going to have some type of protein on its surface, and that protein will sometimes have carbohydrate chains on it. The linking molecule or the linking mechanism for these can be different, but in these specific lipids and glycoproteins, typically we're talking about O-links. So I'm just going to put O-links on here. Now, tumors will increase the expression of the enzyme that does this linking, and so what you end up getting is a protein that ends up looking something like this. Instead of having its normal 3 side chains, it'll have 4, 5, 6, 7, 8, 9. It'll look like a Christmas tree. It'll look like my nose hair. Now, with one exception, these are mostly just used as detectors of things to identify different kinds of cancer. For example, GM2 and G2B GM2 and GD2. I have written here that they're expressed on melanomas. It's melanomas and neuroblastomas, so melanomas and neuroblastomas will express these specific kinds of glycolipids and glycoproteins. CA125 and CA19-9 are mucins found on ovarian carcinoma. And I saved the coolest one for last. The last one is called MUC-1 or MUC1. So what I've drawn for you here, in case you're not a 13-year-old boy and can't pull this off of any image, is a breast. And inside the breast we have ducts, so we have ductal cells. And on the apical surface of those ductal cells, we're going to express something called MUC1, which is a glycoprotein. And so these ductal surfaces never actually get seen by the immune system. Anything that comes in here is getting washed out typically by milk. However, in ductal cell carcinoma, the polarity that is maintained in these cells is lost, and so all of a sudden you can start expressing MUC on the bottom pole of the cell, where it can actually be seen by T lymphocytes and dendritic cells, and it can be processed and presented as antigen. Or so that's kind of the theory, and so there are efforts underway to start using MUC-MUC1 in vaccines. And so I've told you a whole lot of stuff that's like experimental vaccines that are in clinical trials like right now, or as of 2014 when Abbas published his chapter 18 in that book. And the last kind of antigens are called differentiation antigens. And these are basically any antigen that is produced on the surface of the tissue of origin. So for example, PSA is produced by a normal prostate, usually only a prostate and a prostate tumor. CD20 is produced only by B cells and by B cell lymphomas. These typically do not cause an immune response. So our immune system, we will not produce an immune response. However, we can produce chemotherapies that have these as the target. For example, rituximab is a CD20 mouse. It's a modified mouse monoclonal antibody, and it's modified so that it can be used in humans without causing other immune problems. Okay, so the last part of this is to focus on how the antigens get presented to the CD8
positive cytotoxic T cells, because CD8 T cells are the primary T cells for tumors. These are the cells that are actually going to kill other cells. And sort of the dogma is that dendritic cells present antigen on MHC class 2, but I guess fairly recently, it's not been recent, but it's been fairly recently, the concept of cross-presentation was discovered and described. So if I have my tumor cell, of course it's the one in red, and it's got antigen inside of it, and here's my dendritic cell that's about to engulf it. Through various mechanisms not discussed here, it can actually process that and present it on MHC class 1. One of the mechanisms is that it's thought to allow a certain amount of antigen to escape from endosomes and from lysosomes, and then somehow move down the normal MHC1 pathway. Then, of course, the CD8 T cell is going to come and recognize the antigen with its receptor, its TCR, and there's going to be co-stimulation, and the CD8 T cell is going to be activated, and it's going to multiply and cause a whole bunch of havoc. So one of the questions is, what role does CD4 positive T cells have in this? It seems like they're indispensable, but we don't know exactly why. And so here's a couple of, so CD8 are enhanced by CD4, and the idea is why. So cytokines probably enhance MHC class 1 production. So TNF and IFN interferon gamma probably cause the tumor cells. So this is my, so let me make it red because it's a tumor cell. This is my tumor cell. It's going to cause it to increase the production of MHC class 1. Cytokines either from CD4 or from the antigen presenting cell might enhance CD8 activation and enhance its effector and its memory function. And so lastly, remember that CD4 has a CD4 ligand, or CD40 ligand, and the APC has a CD40, and when the CD40 binds the CD40 ligand, then it increases the APC's ability to present antigen. So it will increase presenting of antigen. So what I have here is I have a dendritic cell with MHC class 1 and MHC class 2, so it's probably processing the cell in two different ways and presenting antigen on two different MHC molecules. And then by the CD4 activation from the dendritic cell, we're getting cytokines sent to CD8. So how exactly is this happening and what exactly is going on? So when the dendritic cell binds to CD4, dendritic cell is probably going to send over some IL-12. IL-12 is going to enact the Th1 response, the Th1 phenotype. So this was IL-12. And whenever the Th1 phenotype is induced, then you get the production of interferons and perhaps also IL-21. And just the fact that it's activated, we're going to say IL-2. Now, I'll point out that IL-21 isn't part of the Th1 profile, but the book says that this is going on and it's coming from CD4 cells, so I put it in here. So the second sort of paradigm is that you have the Th1 or the CD4 binding with the dendritic cell, and that through CD40 ligand on CD40, you make that dendritic cell hyperactivated and able to better present antigens to CD8 T cells. So I forgot to put in here that this is CD8, but it should be obvious because it's a black cell. And in this case, the dendritic cell is going to be secreting the cytokines onto the CD8 cell to activate it and to cause it to prevent exhaustion. And so the major cytokines acting on CD8 T cells, on the CD8 T cells, IL-2 to cause differentiation, IL-12 to go from naive to effector, IL-15 survival of the memory cells, IL-21 prevent exhaustion, interferon gamma again from naive to effector cells. And most of these can also act on the NK cells as well. So we've talked about the T cells. What about the B cells? There are some antibodies against tumor antigens, but there isn't really a demonstration that they have an effect in vivo. However, when used pharmacotherapeutically, they do help. They enhance probably through cell-mediated cytotoxicity. So you remember that that antibodies can either cause complement fixation or they can allow the FC receptor of a macrophage to phagocytize a cell. And so it's probably that mechanism where the FC receptor is binding to the antibody that these products are actually working. So typically a tumor will reduce its MHC. So if you have, let's say you have 50 tumors or 50 tumor cells, we'll just draw 5 of them. We've got 5 tumor cells and they're all expressing MHC class 1, except this one was like, I don't really want to express it a whole lot. He got randomly mutated. And then you had CD8. CD8's a real BAMF. CD8 came over here and he killed all of these because they were expressing this crazy protein. But he came over here and he started looking around. He's like, nothing's wrong with you. So all of a sudden this guy, he had a bunch of babies, and all those babies expressed like very little MHC as well. And then, so you had now this new population of cells. And so that's one thing I want to actually just, I think is actually particularly high yield, is that you get mutations that are spontaneous. And this is the big misunderstanding, I think, with whenever we teach monoclonal production of cells, or of tumors. So this is called the one-cell hypothesis. And this is true. This is actually how we believe tumors really work. So you have one cell. That one cell gets a whole bunch of DNA mutations. Boom, boom, boom. DNA's messed up. And that one cell makes many, many daughter cells that are identical, and all those daughter cells are tumor cells now, and they all make identical daughter cells, and they're all tumor cells too. But the problem is, that's true except for one thing. Those daughter cells, most of them are identical. Not all. Not all daughter cells are identical. And so you get some daughter cells that can withstand low oxygen content, and they can withstand hypoxia, and they become anaerobic, and they can survive without blood and vasculature. You get some that can survive the attacks of CD8 because they don't express MHC. You get others that can survive the NK cells because they can secrete various cytokines. And all of a sudden, these accumulations of these immune bypassing mechanisms occur, and eventually we get this tumor that the immune system just cannot fight. And so I said all that to say this, because the NK cells, they identify cells with reduced MHC, and so because of the CD8 response, we are selecting for the mutations where the MHC is reduced, and it's most effective against blood cells. That is, natural killer cells are most effective against blood cells. For example, Epstein-Barr virus-associated lymphomas. You can actually activate them with IL-2 that make them extra active killers. You incubate NK cells with IL-2 and then inject them to somebody that has an Epstein-Barr virus-associated lymphoma, and they're really, really good at killing those. Now, what about other parts of the immune system? Macrophages can have 2 roles in the tumor immune response. The first thing is that macrophages can, by encountering damage-associated molecular patterns and from interferon stimulation, they will use nitric oxide to kill tumors. Those are macrophages expressing the M1 phenotype. There's also an M2 phenotype, and these actually will cause secretion of VEGF and TGF-beta. This will contribute to tumor growth. We'll talk a little bit more about this whenever we talk about the tumor's escape from the immune response, which is the next section, part 2, failure of the immune response. And it basically is 3 mechanisms: specialized tumor mechanisms of evasion, tumor cells resemble host cells, and the rapid growth overwhelms the immune system. So first of all, if we have tumor cells and they are our cells, if they look exactly like our cells, then we can't have an immune response against them. Otherwise, that's called autoimmunity. We will be hurting our own cells. And in fact, some tumors are associated with autoimmunities. For example, thymomas. So one is because they look so much like our own cells, that it's hard to fight them in the first place. The second is that they grow really rapidly, and if you have a really, really strong immune response to it, if it's growing rapid enough, it can outgrow the immune response. It'll just grow faster than it's being destroyed. It's like if I have a snake, let's just say I have a snake, and he's being eaten at this end by a cat. As long as he's like growing faster than the cat is eating, I'm just making this up, but then he'll keep growing, right? And the other thing, the big high yield, is that they have specialized mechanisms of evasion. That's what we're going to focus on for part 2. So I've broken down these specialized mechanisms of evasion into 5 groups. So you have loss of antigen expression. We talked about that, downregulating the MHC. Upregulate inhibitory elements. So these are surface elements and secreted elements. The Tregs also reduce anti-tumor lymphocytes. So the Tregs can reduce your immune response against the tumor, and the Treg's just doing its own job. He's trying to prevent your body from attacking itself. The M2 phenotype of macrophages can help enhance tumor growth and angiogenesis. And then there's something called MDSCs, myeloid-derived suppressor cells, and we'll talk about those. They actually suppress your body's immune response against the tumor. So we already kind of talked about the misconception of the one-cell hypothesis. It's not a bad theory. There's just misconceptions that arise from it, and the major misconception is that one cell producing the tumor means that all cells are identical, when in actuality all cells are not identical. There are different mutations within different cells of the tumor. And I think out of everything, that's probably the most high-yield, big concept I've given you. So the first thing is I have this tumor cell, and it's producing a tumor-specific antigen. So remember, we can produce tumor-specific antigens or tumor-associated antigens. I'm going to say it's producing a tumor-specific antigen, and all of a sudden, you make a whole bunch of copies of this guy, and then eventually you get one that's not making any tumor-specific antigen. It just decided, I don't want to express that one. And so whenever CD8s come over and destroy all these, this guy gets selected for and he survives. And so you can have differential selection for reduced antigen and reduced MHC. The key is that the antigen cannot be necessary for the phenotype. If it's necessary for the phenotype, for example, if it's a P53 antigen, then that would actually destroy the tumor, because you have to have the aberrant P53 for the tumor to keep going. You can't just say, oh, I'm going to start making normal P53. It just doesn't work like that. So the next thing is that you can upregulate inhibitory elements. So you'll remember that CTLA4 and PD1 actually inactivates T cells. It's one of the things that the Tregs will use to inactivate. And then also recall that TGF-beta, when secreted in large amounts, will suppress lymphocytes and macrophages. And so once you have tumors that are both expressing CTLA4 and PD1, that is in your cell's DNA. Your cell's DNA all have it. It's just that you choose not to express it in certain kinds of cells. Those cells can start re-expressing those proteins and reduce the T cell activity. Then they can also upregulate the expression of TGF-beta and cause reduced lymphocyte and macrophage activity. Okay, so the next thing was Tregs. Hey, what I need to say really, it turns off the immune system, and if it makes sense that it's stopping you from killing yourself. That's what Tregs do. They keep you from killing yourself, and they think that cancer is self. So the M2 macrophages, we already said that they secrete IL-10 and PGE2, which will cause T cell inactivation. Or I don't think I did say that. I said that the TGF-beta and VEGF will enhance the tumor growth and vascular growth, but they also secrete IL-10 and PGE2, and both of those have T cell inhibitory activity. And so when those secrete PGE2, those M2 macrophages, it can actually recruit these myeloid-derived suppressor cells, and these are basically just immature myeloid cells. So you have immature dendritic cells, immature monocytes, immature neutrophils that are being pulled out of the bone marrow by various chemokines. And so the one thing that they have is they all have CD33 CD11B, and CD15 as cell surface antigens. And one thing that these myeloid-derived suppressor cells will do is they'll secrete IL-10, and they also, so they gather in the lymph nodes. So they come out of the bone. So here they are in the bone. And they come out of the bone and they go into the lymph node. You'll remember that the lymph node is where you have a bunch of T cells. Inside the lymph node, they start creating free radicals, for example, peroxynitrite. And so the peroxynitrite will come in and kill T cells, and also IL-10 will inhibit the T cells. Another thing is it'll produce a protein called indoleamine 2,3 dioxygenase, which destroys tryptophan. And if you destroy tryptophan, it's needed for T cell proliferation. And so for one of the things that you might do for this, just a simple increase tryptophan in the patient's diet, but I mean, that's still got to be studied and stuff. And then lastly is that these myeloid-derived precursors will actually induce Tregs, and they'll also skew T cells toward Th2. Now remember that we have Th1, we have Th2, and we have Th17, right? So I always like to think of these in this manner right here. So Th1 being at the bottom, it's intracellular pathology. Then Th17 being at the top are extracellular pathologies. And this is just the weird one. This is like the reaction to type 1 hypersensitivities like histamine reactivity. This is where you get reaction to certain kinds of parasites. So it's not that this is bad, it's just weird. And the myeloid-derived precursor cells will cause a shift from the Th1 phenotype to the Th2 phenotype, which has absolutely no impact on a tumor. And part 3 is the immunotherapy and treatment strategies. So you have different strategies based on the body's immune system. So you can have vaccinations with tumor-specific antigens, like we talked about. You can have blocking of inhibitory pathways, so the tumor tries to inhibit the immune response, and you can try to block that the tumor's blocking. So here's the tumor, and he's trying to block the immune response. And you are going to say, I'm going to block your blocking of the immune response, and then the immune response can attack the cancer again. Another strategy is to augment a host with cytokines. Like we said, IL-2 is one of the things that you can use to enhance the activation of certain leukocytes and more effectively kill cancers. This one, inoculation with inflammatory substance, and it requires a little bit of explanation. The inflammatory substance is actually the killed Bacillus calmette-guerin. I'm sure I'm saying it wrong, but basically this activates macrophages and everything to come in, and by the macrophages coming in, they're like really irritable from this specific bacillus, and they're just pissed off and they attack the cancer cell. It's used as an adjuvant for T cell response as well, and it's it's used to treat bladder cancer. So I gave an example of the inoculation with inflammatory substance. Let me go back. The inhibitory pathway, there's anti-CTLA4 antibodies and anti-PD1 antibodies. These are, the problem with these is that they often cause autoimmune reactions. So if you're blocking CTLA4 and PD1, that's what the Tregs actually use to prevent autoimmune reactions. So whenever you're using those antibodies, you have to be careful about overusing them and causing an autoimmune reaction. Then there's passive therapies, which say, hey, I've got some anti-tumor antibodies I'm going to give you, which we've already talked about that in a bunch of different contexts. And then the other is that you can transfer activated T cells, and that has its own set of complications. But one thing you can do is like an autogenic graft. So you can take out your T cells, activate them towards, a biopsy of the tumor, and put them back in and let them do their thing. And then there's the extreme form of that idea, and that extreme form is called adoptive cellular therapy. This is really cool. So let me explain this really quick. So this is something called adoptive therapy, using T cell expressing chimeric antigen receptors, or they're called CAR receptors. So here I am, and needle comes in, get a biopsy. This is a biopsy of my bone marrow, and I'm taking out T cells. So this is my T cell, and I should have made it black, because this is a T cell, right? Next, I'm going to genetically modify my T cell. And so what I'm going to do to the T cell is that I'm going to take and splice a whole bunch of genes together. So I've got a gene, let's say a gene that encodes for a receptor to that specific tumor. So this is a receptor to a specific tumor, and then I'm going to splice that in. And it's splicing using the Ig variable chains. So you remember that they have Ig variable chains, and these can be spliced around in a bunch of different ways. Well, you can splice these, you kind of experimentally splice these until you find something that will attack some tumor-specific antigens. Then you code in a cytoplasmic tail that has various signaling motifs encoded into it. Then you take those T cells and you inject them back into me. Well, first you're going to introduce those T cells to the tumor biopsies and make sure that they're reactive and stuff. And you're also going to soak them in a whole bunch of cytokines to get them really junked up and on crack and ready to go fight a cancer. And so this is what I said here is you have adoptive cellular therapy. This refers to a variety of autograft strategies. And then this adoptive therapy using T cell expressing chimeric antigen receptors. So I've got an antigen receptor. It's chimeric antigen receptor. It's a T cell expressing chimeric antigen receptor. So it's called a CAR, chimeric antigen receptor. So a T cell expressing CARs, and you remove the T cells, genetically modify them to express tumor antigen specific binding site, and then these are encoded by the Ig variable chain genes plus cytoplasmic tails with signaling activity, and they are effective in almost all. Not, I don't know if I can't say all, but I say according to the book it was all, but I think there's been some reports of a few cases that it's not been effective, but it's effective in almost all ALLs and CLLs. And they're coding the receptors to recognize CD19 on the B cells. And if this explanation from Abbas is the same thing that I saw, what I think it is, then they're calling these modified cells, they're calling them serial killer cells. The last thing is another really cool thing, because it's really cool that we're using like a pathology to treat cancer. And this is graft versus leukemia. This is essentially the graft versus host. You get an allogenic bone marrow transplant, and this allogenic bone marrow transplant goes and kills the cancer. So the challenge is that you have to modulate it so that you don't get an actual graft versus host disease. And that basically covers tumors with the immuno stuff.